I'd like to thank the organizers for inviting us to give this DNA talk. We'll be talking about preserving DNA information with human and computer review with a computer system that the Allegheny Lab is bringing on board this year. This is the Blairsville dentist. Blairsville is about an hour east of Pittsburgh. John Yelenic and his bride, Michelle. This is John 4 years ago as a murder victim where he was slashed to death in his home and exsanguinated. This is the Pennsylvania State Trooper Kevin Foley, who was arrested for the crime. He was the boyfriend of the bride, Michelle, with whom he was living at the time. And this is the fingernail DNA evidence from the victim, John Yelenic. And this evidence comprised two people. It was a mixture of DNA. 93% came from the victim, and about 6.7% came as a trace amount from an unknown second contributor. There were 3 DNA match scores in this case. The first was a score of 13,000 from the FBI lab, and a second of 23 million from Dr. Robin Cotton, former director of the Cellmark Laboratory. And there was a score of 189 billion by the True Allele computer system. And so the question is, why are these different? And the answer is there are different interpretation methods. And they vary based on the nature of the data used. The FBI lab uses a standard inclusion method, which does not use the victim profile, as you see on the left, and does not preserve the quantitative data, as I'll show you in a minute. Dr. Robin Cotton's method does use the victim profile as information to infer a genotype, but again does not use all the quantitative data. The computer system makes full use of all the information to the extent that statistical modeling and computation will permit. It did use the victim profile in this case, and it does preserve all the quantitative information present in the data. So this is one of the 13 CODIS loci in the case. What you'll see on the x-axis is the length of the DNA fragment, and on the y-axis is the quantity of DNA at each of these alleles. The 2 tall peaks correspond to the victim, who's over 90% of the 2-person mixture. And then the question is, where in all this data might there be the genotype of this unknown second contributor? And there might be more than one possibility. The quantitative approach is to use all the data. The way this works in this case is the victim genotype is assumed because it's taken from the victim and is seen in all the data. And then the computer will try out every possible allele pair, here at 10 and 13, in every possible ratio of different heights. So it does a massive search on the unknown genotype, on the amount of DNA, on the relative heights of the two, and on dozens of other variables. And in the process, for each hypothesis of what the allele pair would be, it generates a pattern. That pattern is compared against the data. And in this case, we see that the hypothesis of the 1013 predicts the data very well with the 2 victim peaks and 2 unknown peaks. For this to work, the computer has to try out every possibility and assign what its likelihood is based on the data. The FBI lab method, which is the standard method in the US, discards the quantitative data. And the way that's done is that a laboratory-wide threshold is applied to the data so that it's cut off, and instead of having peak heights, you have equal allele events of anything over threshold. Anything underneath would be discarded. And then once you have this reduced data, then any pair of alleles, such as 8-8, 8-10, 8-12, and so on, there are 10 in this case, would be considered to be equally likely. The result of having 10 possibilities of equal likelihood instead of only 1, as with the quantitative information method, is a considerable loss of information. How much? Well, we'll see. So let's look at all 13 CODIS loci in this case. If we look at the likelihood ratio, which is a measure of the specificity of the DNA match at each of the 13 CODIS loci, this is the FBI's result. This is the amount of 10, 20, 30, and so on moving up. And the product of those numbers comes out to 13,000. Dr. Robin Cotton's method, which uses the victim information, obtains more information, particularly at those data loci where there were 4 alleles, as in the case we saw of D7, where you could clearly separate out the 2 tall peaks and the 2 very small peaks. And in that case, the extra information that was obtained by a more informative interpretation approach at these 2 loci increased the match strength 3 orders of magnitude to 23 million, 10 to the 7th. The computer was able to replicate what Dr. Cotton did at these 2 loci, but also at other loci was able to extract more information by accounting for other quantitative effects that are in the data, adding yet another 4 orders of magnitude up to 10 to the 11th, or 189 billion. So one might ask, how is this used in court? There was a Frye hearing and a full day of testimony presenting articles, evidence on these topics. And at the end of the day, both Dr. Cotton's method and our computer method were accepted. This was the first time a computer interpretation of DNA evidence by a statistical program was accepted in court that we know of. And based largely on the DNA, according to the prosecutor, the state trooper was convicted of first-degree murder of John Yelenic. Now, one can ask, how general is this phenomenon? Is this limited only to this case? Is this well known? A study was done 5 years ago by the federal government at NIST, the National Institute of Standards and Technology, where samples were sent out to over 50 laboratories that do DNA testing in crime labs around the country. And what they discovered was that there was a 10 order of magnitude difference between labs that used inclusion methods without quantitative data, getting around 10 to the 4th in the tens of thousands, and the few labs that were using all the quantitative information by eye and calculation of up to 10 to the 14th. So it was well known at that point that there was a 10 billion-fold discrepancy of interpretation of DNA information on the exact same data. Collaborating with NIST, we undertook a study where they synthesized mixtures of known composition, 5 different mixture proportions and different dilutions. And you see those 40 experiments as dots on the scatter plot here in blue. The x-axis is the logarithm of the amount of unknown DNA, and the y-axis again is information measured in log likelihood units, 10 to the 3rd, 6th, 9th, and so on. And what we found is that with computer interpretation that used all the information, down to about 100 picograms of DNA, you would get all the information. And then there'd be a linear decrease down to about 15 picograms of DNA to a 1,000,000 to 1 match score. Human review was shifted 1 entire order of magnitude to the right, and below 150 picograms was not getting much information, which is perhaps why the current guidelines discourage crime labs from looking manually much below 100 picograms. Interestingly, this experiment was used in court, particularly at the admissibility hearing, to show that based on the amount of DNA under the fingernails, we would predict the match score that was found in the range of 100 billion, as opposed to what alternative methods would have found, which would have been very little. We then did another study. This is with the New York State Police. I should mention the previous work was published in December in PLOS ONE, which is an online peer-reviewed journal. A current study that's under review at the Journal of Forensic Sciences And here we looked at 8 cases of 2-person mixtures. Each mixture has 2 contributors, and in orange, you're seeing the information content from 8 cases that the New York State Police had reviewed using inclusion, and on the identical data, not assuming a victim, but just using the information given. In blue, you see what the computer result is. And on average, the human review produced a result of about 10 million, 10 to the 7th. The computer review was 10 trillion, 10 to the 13th. And you see about a 10 to the 6th, or a million-fold information gap on cases that the New York State Police had solved. The New York State Police and cybernetics then took yet another look at the data. And here we ask the question, instead of how much information was obtained by computer, given that a person was able to solve it and put a number to it, let's just look at all the cases. So here what you see on the x-axis is a listing of all 85 cases. The y-axis again shows information in log units, 5, 10, 15, and so on. If you focus just on the large blue background, that's what the computer did. The height is the information, the logarithm of the max score at each case, and it's done in descending order. The median value is 10 to the 15th, or quadrillion to 1, and you get a meaningful result for everything. Maybe there's a few that are under 1 million to 1 at the very end on the right of the sort. By comparison, this is what human review was able to achieve. The different colors correspond to different methods based on how difficult the case was. The 4 gray bars are assuming the mixture came from one person because there was a clear major contributor. The green bars assumed that you could use the victim, and the lower orange bars was human review where the victim was not used at all. And there are 2 notable features of this particular slide. The first is, if you look at how many of the cases where a match score was produced, did a human review produce a match score? The answer is less than 30%. This means that over 70% of the DNA cases, of the DNA items of evidence, there was a usable information that could identify a suspect, but for which nothing was reported, and this was used to, in fact, show that the productivity is such that 3.5 times the amount of work is being done that needs to be if you use human review instead of a more informative computer review. The other interesting concept from here is if you look at the total area of blue, that's the total amount of information over the 85 items that was obtained. If you look at the area of the colored bars of human review and look at the ratio, it's only 20%. So overall, 80% of the information is discarded by human review that computer review actually preserves. So we draw some conclusions from these described and ongoing studies. The first is well known and is published almost monthly in the forensic science literature, is that DNA interpretation of challenging samples, mixtures, low-level property crime, handguns, and so on, varies greatly across laboratory, jurisdictions, countries, and so on, typically 10 orders of magnitude depending on the methods used. Human interpretation discards data by imposing limits, saying what data can be used, above what level, what can be thrown out, what can be kept. Computers, with extensive computer modeling of the quantitative data and modern statistics, can preserve far more of the information that's present for identification in the data. The average loss between the standard threshold inclusion methods and quantitative methods is at least 1 million to 1 in the match score, which may not affect a case where the human result is 1 million to 1 and the computer result is 1 trillion to 1, but when the computer result goes down to 1 million to 1, humans have almost nothing to say about the results at all. So the message for medical examiners is that when you do your excellent scientific work of collecting the evidence and send it off for DNA interpretation, when the answer comes back inconclusive, that might be entirely an artifact of the human review method that was applied to it and not representative of the information that the data contain. Thank you.